So Flic Gabbay, she recently took office as the president of the Faculty of Pharmaceutical Medicine and was elected to drive more interest in the treatment of infection, investment in women's health, which I thought would be interesting to highlight because both really underrepresented areas, and to support innovation in and raise standards of pharmaceutical medicine. Flic is an active fellow of the Academy of Medical Science and is passionate about bringing pharmaceutical medicine into mainstream medical science and its application to public health. She is also the funding partner of tranScrip, a leading life science consultancy, partner of One Nucleus, and a returning supporter of our conferences that we are delighted to have again today. She has spent 4 decades in drug development and over the last 13 years has led tranScrip on multiple innovative projects and drug registrations for over 250 clients. So I think that is setting the high standard for the discussion now. Flic, over to you.
Thank you, Aileen. That's excellent. I hope I can live up to it.
I'm sure you can.
Welcome. I'm going to remove my mask here, I think. And I'm talking to you really from that basis, but I think we've been very close to having a pandemic several times before, and we might have had actually some quite serious viral illnesses that we didn't see before because we didn't have a diagnostic test. So I think that this is, for me, an extremely sad but very valuable exercise that we've been through in the last 18 months, and I think we should certainly learn from it. In addition to the kind words that Aileen said, I did start in big pharma. I was CEO of a couple of biotechs, and I was founder chair of another biotech. So I am well used to both aspects of big pharma and smaller pharma coming into this, and the constraints that apply actually on both sides. So what I'm going to talk about is why COVID as a disease is different from some of the other things that we do drug development for, what research and innovation we've done, and how some bits we've done really well, and some bits we've got a lot more work to do, but it has illustrated where we might do it. I think we have a lot of work to do in terms of application to clinical practice, and we're talking earlier about stem cells and gene therapy and so on. That's going to be a real challenge for us in the future, as was adapting clinical practice and standard of care during COVID. I'm not really going to talk a great deal about collateral impact. We will talk about it in the panel, and I'm just going to talk a little bit about learnings for things other than COVID-19, because actually we have quite a lot of things to learn. And one of the things I'd like to reflect on here is that, I don't suppose many of you remember, but in the late 90s, Richard Bax, who's an anti-infective bacterial person who brought a lot of currently used antibiotics to the market, and I wrote a paper, 3 papers actually, for Scrip at the end of the 90s, saying that the one thing that was missing in the drug development arena was doing research on methods of research. And we still are questioning whether actually using p-values and that sort of thing was the right way to go. And of course, with big data, we are challenging that now, but maybe we should be challenging that with smaller designs as well. So, what was unusual about COVID? And I'm afraid my slides are very busy, but they're going to be available to you afterwards, so I won't necessarily read everything on them, but do just mail Aileen if you would like a copy. Well, one of the biggest problems that we all know about is it spread very fast. We needed to treat people, if we had anything, either with a vaccine or anything else, very fast in a very short space of time. The other thing that actually is similar to all infection, but very different from most of the drug development that we do, is the length of the illness. These, people always said, why do you do trials with antibiotics in an infection? Because the endpoints are really easy. The patient either survives and gets better within 3 weeks, or they die. What's difficult about that? And of course, what is difficult, I will show you, is exactly that. It is the fact that you have a short space of time to intervene and make a difference, and if you don't, they do get very ill. And consequently, we had high mortality and morbidity. We did clearly need diagnostics because obviously you have to treat these patients very early. And the other thing that we have done, and there are people on the panel who might reflect on this, that in the last 20 years, most of our infectious diseases consultants have been either diverted to tropical medicine, or we just haven't got anybody going through infectious disease. And we have a lot of infectious disease epidemiologists, but specialists in treatment of infectious disease have largely disappeared. So when we needed a SAGE, we were struggling for respiratory physicians, for infectious disease physicians. We had 2 people who were ex-leaders or leaders of Wellcome and so on. So they were more general people and microbiologists and tropical diseases and epidemiologists. And actually the drug development piece initially, and that's largely where the vaccines task force came from, was just not represented. And I think that made COVID quite difficult because we suddenly had a very serious disease with a lack of the infrastructure in terms of the academic community and the clinical practice community to deal with this. Also, we now have, compared to swine flu, one hell of a lot more political interference, media, everybody knows everything that happens. For those of you who didn't know Plan B was coming at 6 o'clock last night on Tuesday afternoon, then you weren't listening or you weren't reading. We know everything that's going on, and that made this pandemic very different. We also have actually, and I think the ABPI have done an enormous amount of work recently on educating children, but public health, clinical medicine and drug development had not been part of a healthcare professional's training in the same way as it probably should have been. And now, ironically, we have teenagers who come and ask us what is phase 1, 2 and 3, but some of our junior physicians have not any idea what the pathway of drug development is, because we don't have it in medical schools, except 1 or 2 that Sheuli's been working on, but we are actually slowly getting it into school curricula. And I think it is really important, because if we have a crisis like this, we need people to know what needs to be done. So enough of the politics and so on. Let's just move on to the disease. This is something you all know. I'm not going to stay on this for any length of time. Obviously, the reason that the virus is so successful is because 30% of the people who are infected are asymptomatic and therefore can act as spreaders. The original SARS in China and MERS, avian flu, although I hate to mention this because I believe we've got it back again, but avian flu in humans had a 30 to 50% mortality. Not much good for spreading. You've killed off your vectors. So COVID-19 was extremely good at that because it could be easily spread, and I think Omicron is going to be even better at it. The overall mortality, actually, overall, if you take all age groups, is quite low. But we had this huge problem with elderly people, and of course, because of ACE2, I'm afraid you guys in the audience have less ACE2 receptors than us women, because so we can compensate if they're blocked by a virus. because it's controlled in an X gene. So there you go. Man flu exists. Or at least you've got an excuse for it now. So we knew a lot of this right at the beginning. And we also knew what was in the toolbox. We knew that vaccines were really important. Now, if you look at this triangle, the triangle represents the breadth of spectrum across viruses. So in terms of the different variants, on the whole, although it appears to be being proved slightly differently here, because it looks like the Pfizer vaccine does work against Omicron, but on the whole, vaccines are quite narrow spectrum. They will not do things. Passive immunity and our monoclonals are a little bit broader spectrum, so we have Synagis and the RSV monoclonals that we knew would work there. Small molecule antivirals are broader spectrum too, and as you can see here where I'm going, we were able with the antivirals to be able to sort of divert and repurpose antivirals that had originally been in development for something else, Ebola in remdesivir's case and flu for molnupiravir. And then we have the host-directed treatments, which are much broader spectrum and which we probably should have spent a lot more time on in the past. There's a lot of papers on steroids in ARDS and respiratory failure. There's a BMA guidance actually saying that for people who have ARDS after antiviral treatment, that they probably should be on steroids. So actually, the good thing about the dexamethasone story is that it actually underpinned that absolutely, and of course that's been a very valuable thing to understand. But tocilizumab was already on the market, and the passive immunity mAbs were not repurposed, but we did know well how Synagis works, and so it was an obvious area to go. So from this slide, my main message is, let's remember that actually there are host-directed treatments here that will be useful for other viruses, not just other variants, but for other viruses. What about the clinical development aspects of this? Well, the vaccine trials and the vaccine story is a huge success. It is a success because of the task force who got everybody together and collaborated, and pharma and governments worked together, invested invested in the technology. The vaccines, the initial vaccines we have, the Pfizer one and also the AZ one, were sort of repurposed. Obviously, a lot more work was done on them, but the technology was there and was, in Pfizer's case, was being used in oncology. That was where it was destined. So, but we had endpoints. We know if you give a vaccine, we don't want patients to get the disease. We don't want them to be hospitalized. We don't want them to die. We can count that from healthy people. So if Steve Lockhart was here, and unfortunately Steve Lockhart from Pfizer has been banned to travel and we couldn't get him on the screen, so he's not here. But he would say he looked after the Pfizer vaccine, and he would say that the tried and tested vaccine development pathway worked. It was a huge amount of work in the space of time it was done. We've never done it that quickly before, but it did actually, it was quite successful using the methodology that we already had in place. So we shouldn't really be throwing out the rule book altogether. Supply chain was critical, thanks to Kate Bing and a lot of other people. That was brought very much to the fore and very important. And June Rain, I think, did a stunning job in getting stuff through the MHRA for us. And of course, we all know the stories around the world where things went a bit more slowly. So I think it was an amazing story. I think in the mild PCR positive pre-hospitalized patients who are eligible for passive immunity, that's another good story. These are people who are vulnerable, who you want to treat with an IV because we can't treat everybody with an IV. And you can reduce the viral load and the illness. And of course, Southampton last week, and before that, the Regeneron Roche projects and so on, they reduce hospitalizations down by about 80%. However, these are hospitalizations in vulnerable people who would be expected to be 7 or 80% only. And we could not use those for everybody who has mild symptoms because we just couldn't do that as a country. And so those are for vulnerable people. The new ones are more broad spectrum. The early ones, Lilly, of course, was withdrawn from because it didn't have any effect on Delta. So now the challenging bit. There had to be a sting in the tail a little bit, and this is the same problem we've had with infection for many years. Hospitalized patient trials, those patients with severe COVID who are hospitalized, is a real challenge. The deltas are smaller. The difference between placebo and active is much smaller. Mortality is high. They're confounded. Almost all patients who are hospitalized, not all, but almost all of them have some sort of confounding illness. So we have lots of patients in our trials who have other conditions that actually will probably have more impact on the outcome than the treatment that we're giving them for COVID. The other problem is that a variation in standard of care was really considerable around the world. And in some of the studies, and I think, Ratko, you're sitting here, Ratko and I work on a Synergen trial for interferon beta. It's in 17 different countries with 112 sites. And all this is on clinicaltrials.gov, and many countries have completely different standard of care. And when they're hospitalized, whether they give oxygen, what concomitant treatments they give, it makes doing trials very difficult. And we have to do trials in that number of sites because we need that number of patients. And finding patients is really difficult. I call them whack-a-mole trials because there will be, if you have too many patients in the ward, they won't do trials because they can't. It's just too difficult to fill in any forms or anything. If you have too few, you don't have any patients. And with the waves of COVID, you either have no patients or far too many. But occasionally, you get exactly the right moment in time, and you have to be in there. But of course, when we're doing trials, it takes us 3 months to get the approval, so AZ called their trial Storm Chaser. We call them whack-a-mole trials. But it's really, really hard. And I know some people have been looking at different trial designs. We'll talk about that in our panel. So this is really difficult stuff. And I will go on to talk about what that means for other trials in infection, and particularly antibacterials. Also, we mustn't forget we have had some problems with supply chain, both for tocilizumab and for dexamethasone, and that needs to be remembered. Outpatient trials, some of the same things, but not quite so difficult. And as we know, molnupiravir was pulled from the hospital trial because it wasn't going to be able to show a difference, but obviously did in the outpatient setting. So I think what I'm saying here, the message I hope that's coming over, is that it's not easy to do these trials for this aspect of COVID. I've got a couple of illustrations here of that sort of thing, and I am not going to dwell on this very much, but here we have tocilizumab studies, retrospective studies told us, modelling from the databases, that it was likely to work. About 100 clinical trials started, most of them never completed, and when they did, they gave completely conflicting results. On occasion, there was a trial in Brazil that showed that had to be stopped because the tocilizumab patients died more frequently than placebo, and yet there were other trials that showed that it clearly worked. So very confusing and very difficult. Recovery, of course, showed that in a large number of patients, I think the initial study didn't show very much when they did the interim result in July 2020, but when we got to spring 2021, it showed that actually there was a significant difference, but the difference was between 31% and 35% mortality, which is really not huge and is very difficult. These studies were all confounded by giving dexamethasone, by giving remdesivir, by giving monoclonals. Very, very difficult. And the other thing, this is from an editorial in The Lancet, recovery had to be put in place very quickly and is a relatively good study design for that sort of thing, but it's an open study design. There was very little information collected, and it made life very difficult. And on top of that, about 15% of the patients didn't actually get the drug they were allocated to. And that's the difficulty of doing those kind of studies. This is a paper that both Ratko and I authors on, which is the interferon beta phase 2 study. And this is just up here to illustrate that there is a different endpoint that you can use other than simply progressing to mechanical ventilation or mortality. And this shows that at 14 days, patients who are recovered had double the chance of recovering on interferon beta. And inhaled interferon beta is in phase 3. It is also being investigated by NIH in outpatients. So it's looking at the same group of patients, but in a different way from simply mortality and morbidity in terms of mechanical ventilation. I am beginning to run out of time, so I'm not going to spend much on application to practice, but I leave you with this slide because I think it's very important. Standard of care and changes in standard of care for all COVID patients was very difficult. I wish I had printed off the NICE guidance every week for the first 3 months, because it was rapid guidance. It changed. That's the nature of rapid guidance. It's very confusing. And also the issue of sort of evidence base has to be dispensed with. It had to be. We had to give a best guess, really. And I think maybe there is a review of NICE and so on coming through. And I think for me, things like stem cells and gene therapy and stuff like that's going to be very difficult. We think we have a hard time with not enough infectious diseases physicians. We're going to have a much harder time with people experienced with ATMPs. And I promise, and I know you can't really read this terribly well, this is 2019 causes of deaths, published in 2020 by WHO and the UK. I didn't put deaths now because they are masked quite a lot by COVID. But if you look at our top diseases here, you will see that ischemic heart disease and stroke which are really cardiovascular things, still top worldwide. And if you look on the right-hand side in the UK, you might say, well, that's all people outside the sophisticated nations. It's still up there as a major killer, and some of dementia will fit in that box as well. But next, what do we have? Chronic obstructive pulmonary disease and lower respiratory infection. And actually, you could argue that quite a lot of our oncology deaths are due to infections as well. We have to tackle respiratory infection generally, not just COVID. It is so important, but has a lot of analogies to COVID. Short-term disease, the hospital-acquired pneumonia, ventilator-associated pneumonia, high morbidities. Many of you in this room, I'm sure, have had relatives who have had hospital-acquired pneumonia, gone into ICU, got sepsis and bacteremia. It's a very sad reflection on us that we are not managing this, and doing trials in this area is just as difficult as the severe COVID patients. It's all confounded by comorbidities, and it's very, very difficult to work in. Undoubtedly, we could get better vaccines into this space in terms of antibacterials, but I think doing some better style trials in respiratory infection would be really good. I also think that diagnostics are vitally important, but not necessarily the way we're looking at them. Most patients with community-acquired pneumonia do not have a spit, so you have nothing to culture until they get bacteremia, in which case it's probably too late. So it's a really, really difficult area to work in, but please invest in it because we need to go there. And if you get something that helps us as a world, then I think will be really, really valuable. Stroke and heart disease, well, we have the international stroke trial running. We have some really interesting things coming through here. We divide some of the acute strokes. So I think there's a lot of analogies here that actually we probably could learn in terms of prevention, in terms of treatment, and in terms of trial designs. So I think there are things there that we can definitely follow through on. Now, what do I think were the learnings? As the last two slides, I think it was said this morning, it has to be said over and over again. We have pillars of excellence in this country and around the world that actually are very good, but often we don't work as a team, and we need to respect and work as a team much better. And so I think coming from the collaborations in COVID, I really hope that actually does carry on from there. I think the understanding of disease is really important. I think we haven't done enough yet on COVID-19 as to how we understand that. And I think that repurposing is also something we might do a little bit better. I often describe repurposing as though, a car today has a chassis and four wheels. A car in the 1950s, and even in the 1930s, had a chassis and four wheels. But the way we build it and the way we deliver people from A to B is completely different. And actually, there are quite a lot of drugs out there that could jolly well do with being delivered slightly differently. And that's not been looked at enough. And actually, there are a lot of barriers around it that I'm well aware of in terms of IP and so on and so forth. But I think maybe that is something we could address, and the learnings from COVID for repurposing have been quite valuable. Clinical trials, well, I've said enough about that. We've got so much more work to do. Supply chain, absolutely. And I think that new therapeutics generally, I think vaccines are going to be critical. We are going to look at more joined up thinking, I think, which has come out of COVID, between digital apps, medicines, devices, all these things. I've always had an idea that somehow in infection, your temperature does not remain up. It goes up and down. If you monitored people's treatments by measuring continuously their temperature, that would be one simple thing we could do in some of the pneumonia studies so that we didn't miss the peak in fever, or actually also recognise that patients, elderly patients with pneumonia, drop their temperatures rather than peaking. And so there's a lot of work we have to do. And I'm going to be quiet now and bring up the panel members, but thank you for listening to me and my polemics. But I think that we've got a lot to learn from COVID, and I think we've learned a lot. We've done fantastic things. Let's keep it going. So we have a panel here that has been in different ways very deeply involved with different COVID trials. It'd be hard not to be, actually, in the last 18 months if you're working in pharma. And what I'm going to do is to ask each of the panel members to introduce themselves for a few minutes, and just to point out which are the most important things that you want us to elaborate on in the panel. We have people who are in completely different walks here. We've got Aiden, who's a statistician, who will introduce himself in the middle, at the end, and we've got Professor Ratko Djukanovic, who has been heavily involved in a lot of COVID, both from the perspective of NIHR and from Synergen, which is a biotech spin-out of Southampton, and Sheuli, who's been incredibly involved in all the national things and has just sat on the clinical trials guidance for some of the collateral damage to other trials that COVID has done. So, Aiden, do you want to start introducing yourself, and we'll come along the panel that way?
Yeah, thanks, Flic. Hello, everyone. It's great to be here. So, I'm Aiden Flynn. I'm the founder and CEO of a company called Exploristics. We work with pharma and biotech companies to help them with their clinical trial design, to make sure they're running the right studies, they're collecting the right data, and then we'll support the analysis and reporting of the data as well. So I've worked in the industry for close to 30 years now. I started in academia, then worked at Glaxo for 10 years and set up Exploristics 12 years ago. And as a statistician, I've been able to observe some common practices in the way clinical trials are designed and conducted, and I think that many of the clinical trial failures could be avoided through, and you won't be surprised to hear me say this, through much earlier engagement with statisticians. Because, it's really the statistics that help to de-risk the clinical trials, that make much better use of the available data at the design stage, and that earlier engagement would really help with that. And in particular, I'm a big fan of modeling and simulation to evaluate lots of different design and analysis options to make sure that the studies are set up for success. So I guess just with the COVID era, there are a few things that I think have been quite helpful. One is, I guess, the very public use of modeling and simulation to inform decision making at government policy, and I think that has actually fed into some studies that I've been working on in terms of clinical trial design. So I've seen a much more open use of modeling and simulation to guide decision making, which has been great. The other, I think, thing that has been useful, I know we've talked a bit about big pharma, and maybe it's quite a conservative, risk-averse beast. And for any of those out there who've tried to sell products and services into big pharma, it is risk-averse. You've got to jump through lots of hoops in order to sell your products and services, even though you've got a clear value proposition, and even though you may believe it's a no-brainer. It is still very tough, and I think there's been a bit of a shift within big pharma in the last couple of years, where maybe they're less likely to put up those barriers. I think they've moved from a position of, well, this is the way we do things, or this is what our process says, to one where they ask, okay, well, why do we do things in this way? And how can we do it better? Why does it take so long to develop vaccines? How can we do it quicker? That sort of thing. And that's been useful because they've been able to adopt more technology in their study designs. And I think some of that is going to stick. It's given us an opportunity to learn about what works and what doesn't work. And I think that has been very useful. And just the last point that I think has been very useful, and it's around access to data, access to good data to help drive decision making. Any modeling and simulation has to be grounded in some sort of reality, and to do that we need access to data. So I think there's been a real push to try and get some early access to good quality data to help drive some of that informed decisions.
Thanks. Ratko.
Right, well, first of all, thanks very much, Flic, for inviting me. It's a real pleasure. You and I have known each other for probably too many years that we would want to declare. So thank you. I've actually got quite a long list. So I've got an ed memoir. So who am I? I'm the man whose surname is difficult to pronounce, Djukanovic. I tell them, think of Djokovic, then take away the millions that he has got. I am a professor of medicine in Southampton, but I'm a chest physician by sort of clinical training, and my interest is in chronic airways diseases, in particular severe asthma. And I also have I won't say a hobby, but nobody wants to do chronic cough. So I self-selected myself, I guess, and took on this. And I was very much in demand during this COVID, and I still am. Floods of patients coming in, because if you cough, everybody thinks you've got COVID. I'm also the theme lead in the Biomedical Research Center in Southampton for respiratory and critical care medicine. So really, I couldn't have been in a better place than I was at the time of the pandemic. And I remember a first phone call that I got from Jonathan Sheffield, who was the chief executive of NIHR Clinical Research Network, who said, "Oh God, I was hoping to retire," and he got a phone call and said, "Come back, we need you." And so I've had the very good fortune to have been party to a number of developments, and I can't praise enough, the NIHR as a fantastic institution, which I think is second to none in the world, and of course the Medical Research Council. And And what I was also privy to was to have helped establish the so-called translational research collaboratives, one in respiratory medicine. So we have in this country all the ingredients, the structural ingredients to really undertake first-class research. And because it was there, I think this is why, you know, we got into action literally within weeks. And finally, as Flic said, you know, I'm a co-founder of Synergen, which is a small biotech company, and we're developing interferon beta for viral diseases, viral respiratory diseases, and we had a very successful phase 2 trial. So what are the things that I would like to, I have a long list, I'm not gonna, don't worry. But
We'll let you come back to them.
the first thing that comes to mind is, and I think it goes, it follows on from what you said, Flic, you know, the methodology, we have to understand, we have to do research into the methods of research. And so I have many ideas, and I'm sure you all have many ideas about what could have been done better and what should be done better next time round. But what my main conclusion from all of this is that we really need to apply ourselves scientifically. We need to look at how research was done. In other words, we need a review. It has to be independent. It has to be independent of NIHR, MRC, governments, and so on. It has to be really forensic, using statistics, you know, good statistics to really dissect out the processes that we had put in place. The second big sort of takeaway that I've come with from my experiences is the power of collaboration. And it was really truly wonderful to see what was set up. So we had the urgent public health panel, of which I was privileged to be part of, which really oversaw the approval and implementation of all the trials, many trials, thousands, hundreds of thousands of patients going through them, including the platforms like Recovery and REMAP-CAP and Principle. And so I cannot strongly enough make a plea for collaboration. And we heard about the risk that pharma has to take. And I think this is where governments need to partner with pharma. And my understanding is this was done correctly with the AstraZeneca approach and the Oxford AstraZeneca vaccine. And you need to de-risk, because if those trials had failed, well, you know, so would have that would have been the end of the chief executive of AstraZeneca, I suspect, and the same would have applied to the Pfizer one, because these are expensive studies to do. So really, this is where I think I'll stop, and I've got loads of other things that will possibly pop up.
Very valuable points. Sheuli, I think you come at things from yet another completely different angle.
Yeah, thank you, Flic, and thank you for the invitation today. So my name's Sheuli Porkess, and I'm founder of Actaros, which is my consultancy specialising in policy and medical affairs. But I do a number of things. So I advise to C2AI, a healthcare analytics company, vice president of the Faculty of Pharmaceutical Medicine. I'm on the steering committee for the UK Pandemics Ethics Accelerator, and I'll come back to that. And also, previously, through the pandemic, I'm the former Director of Research, Medicine and Innovation at ABPI, and I also was sitting on a couple of expert panels that fed into the DHSC COVID therapeutics. So sort of a number of angles there. And I think for me, there are two key things, two sort of sets of learnings. One is how to make better decisions, and the second is about how to embed patient focus. And I know we've touched on patient focus already this morning, and it's an area that lots of organisations talk about, but I think it's really sort of been been thrown into sharp relief, as many things have by the pandemic. So on decision making, I think there are three things. So it's relevant expertise, again picking up on a point that Flic made earlier. Having the right structures in place to enable those decisions to be made, and also how to bring in the ethics. And the sort of the insights to share there are, on the relevant expertise, we, as the Faculty of Pharmaceutical Medicine, as FPM, did a piece of work last year to look at the impact of the pandemic on clinical trials and what, you know, our members would say needs to be, what the learnings were. And a number of things came out, including, as you can imagine, better risk planning to be built into clinical trials. But actually, what also came out was the importance of having expertise in pharmaceutical medicine and medicines development embedded within public sector decision making. So this report came out a year ago, and then actually 3 weeks ago at the Faculty Annual Symposium, we had both June Rain from MHRA and Professor Jonathan Van Tam, both highlighting that point and speaking really passionately about this need for cross-sector collaboration and skills and knowledge transfer. So that need for expertise in decision-making is number one. The second bit about having the right structures might sound a bit processy, but actually you need to have the right people in the room to enable that decision-making. And I was chair of the advisory group to the UK Clinical Research Recovery, Resilience and Growth Programme Board. I'll just call it RRG from now on, which sounds like, you know, oh yeah, that's yet another committee. But actually, when we saw many trials being paused or halted in non-COVID conditions 18 months ago now, I was involved in work to try and get those trials restarted at a, you know, across the UK level. And actually, what you needed was some really in-depth conversations, some very challenging conversations across the life sciences ecosystem, and we needed to set up the structure that both allowed those conversations, but also then allowed the sort of the development of an ambition for clinical research, and then the conversation to say, okay, if that's where we want to go to, how do we get from where we are with lots of paused studies to there? And there have been a couple of documents come out from that group this year. And yeah, we continue to work on, you know, what is it actually clinical research should look like. Third piece on decision making is ethics, and I said I'd come back to the ethics bit, and I think we've seen a lot about ethical considerations sort of coming after the decision making, and actually how do we bring that thinking in before you make a decision that's affecting populations. And the UK Ethics Pandemic Accelerator is a UKRI-funded initiative to work to bring ethics expertise to bear on the questions that we're facing now, but also think about how do you build that for the future. And so that's on decision making. So three things: expertise, structures, and ethics. On embedding patient focus, this is something people have been working on for years and years, and I've focused on patient involvement in clinical research in sort of various things nationally and internationally. And I really thought we were making progress. If I'd been here 2 years ago, I'd have said, yeah, patient involvement in research, we're really getting somewhere. And then we've got loads of guidance, international collaborations. The pandemic hit, and one of the things that really happened was researchers weren't involving patients in their designs of trials. So HRA reported that normally they'd have of their applications for ethical approval for clinical research, 80% of applicants would say, yeah, we've either involved patients or we're going to. The first 40 applications for emergency COVID trials that came through, only 22% had done it. So really that sort of drive of let's get patients involved in the design so the protocols work and the results are more meaningful and all the rest of it just dropped. So that made us as a community all sit up and go, oh my goodness, right, we thought we were getting there, we've got to go back to basics. And actually that's on a backdrop of lots of populations being routinely excluded from trials, so women of childbearing age, pregnant women, older people, you know, the list goes on. And we'd all known that as a community, but then that was absolutely highlighted by these disproportionate effects of COVID on different communities, where you say, actually, we're doing trials, but excluding loads, but the diseases affect them differentially. What does that mean? And I think, I mentioned I work with this company C2AI, and they were able to run their algorithms and actually highlight the different effects of COVID on different populations very early on. And I think to Aiden's point about, how do we use that data and those things that weren't available to us before, how do we actually use those proactively more and really look at these different populations and work out how we're going to reach out to these underserved communities, because whatever we thought we were doing before, and I count me in that week because I really thought we were getting there, actually it didn't stand the test of the pandemic. So more work to do on, when we talk about patient focus, actually taking it to a proper implementation level of what are we going to do tactically, not just nice words on a PowerPoint. I'll stop there, Flic.
Okay, I think all of these raise quite interesting and very challenging topics. One of the things about all of the sort of stuff that you've been talking about, and also within the research community, is the communication that we manage throughout the ecosystem. And I think that is very difficult because we can have policy decisions, but just like we don't necessarily have enough infectious diseases physicians or respiratory involvement in drug development, we also struggle to deliver that message to applying research to practice. And I wonder, maybe, Ratko, if you could talk a little bit about communication. There were a lot of things that fell off the side of things when our normal system. One of them was, of course, we had lots of non-peer-reviewed information, and then I'll come back to both of you about communication as well. But do you think that helped or didn't help, or because it was very confusing getting many, many publications, and some were of trials that possibly should not have appeared, she says. I'm not going to mention any.
Yes, well, of course, there's always a risk. There was a certain attraction to about the novelty of various studies. Anything that had COVID on its, in its title, was given priority, and the two world's most important journals, New England and Lancet, essentially turned themselves into COVID journals. Yes, I think the risk, of course, is that things are done in haste. But I think if this is why it's terribly important to have all the details, all the data, the statistics, and so on, that is provided in sufficient detail for people who want to scrutinize, actually are able to do so. And there's nothing wrong with, going back and kind of reassessing and possibly even rewriting certain papers. I think this could be done, and I think should be done, in order to identify, because you only get, a journal will typically want to know, your key messages, and of course the things that are positive get given prominence. But I think what's equally important is what didn't work, so that we see the remaining unmet needs. So, but thank God that we have, the internet. Can you imagine where it would have been without the internet and without the speed that it provides? And the final, just sort of, not word of caution, because everybody is, I think, very much aware of this, that you can get certain groups that then hijack the information and turn it into misinformation. And that's an enormously big risk to us.
Yes, and I think there's quite a lot of work looking at that, and I know the Academy has got a work stream looking at, the Academy of Medical Sciences, looking at how we communicate with journalists and so on, because I think there was inherently in pharma, we have so many constraints. I mean, all of us, probably including most biotechs as well, we're all terribly nervous about saying anything in public, about anything, until we get more or less to the end of phase 3. You know, it's an absolute no-no. But when we started collaborating with academia, we suddenly found things that we put away that we wouldn't talk about suddenly popped up on the 10 o'clock news. And I think that was quite challenging to deal with. So do you think, just going back to, do you think there's a way that we can constrain or come up with a policy that is has some sort of equipoise between the sort of pharma approach, which is don't say anything, to academia, which obviously academics need to say things because it's important for their departments and everything else?
I think because of the commercial aspects of pharma, I don't think this will change, and nor do I think it necessarily has to change. I think the duty is to to the company, to the shareholders, and so on, is to de-risk in respect of misinformation or too early information. But so I think if you have this is where the public sources of information, academia, I think, could make up for the lack of industry insight into what's going on. So I don't think things will. I can't see, Flic, how one could change.
Just get more collaboration and working together, and that sounds. Aiden. In terms of communication and what was in journals and.
Well, I mean, I just remember back as the pandemic started to really take hold, and trying to design clinical trials back then with very little information on which to base the design or support the design, and scrambling around looking for information about variability in different inflammatory biomarkers in a COVID population, or what was the average length of stay in hospital or ICU. Those sorts of statistics in the early days were quite difficult to get hold of. And, there was a nice publication came out of China and then one from Italy, where you could start building a rationale for a study design. But it sort of showed the importance of some of the early release of data, whether it's peer reviewed or not. Personally, I felt it was important to get it out there, and then somebody like me or anyone working in science and clinical trials can judge the credibility of that data and treat it accordingly. But I feel that it was important to get it out and let other people do what they want.
And I would agree with you, and I think, and I'll come to you in a second, Sheuli, and obviously on the Faculty of Pharmaceutical Medicine website, very quickly we put up a review of the whole, everything we knew, really. But I think a lot of companies, and I know talking to other companies, produced a sort of Bible of what was coming out and just added to it all the time. And I think, we'd normally do that over a year. Suddenly we had to do it over 6 weeks. It was really challenging. Sure.
Yeah. So I think, sort of just building on that, I think work of organisations such as the Science Media Centre, which has obviously been going for a number of years and brings together media and experts. And I mean, their work over the last 2 years has just been brilliant in terms of trying to sort of say, okay, this stuff is going on in the world. How do we bring experts to explain the significance, whether that's a preprint publication or a major policy development? And I think that's been absolutely brilliant to help scientists have a voice and being a resource for, journalists. I think, building on that's really important. I think, the other thing I would say is, I think, the work of organisations such as the Science Media Centre, which has obviously been going for a number of years and brings together media and experts. And I mean, their work over the last 2 years has just been brilliant in terms of trying to sort of say, okay, this stuff is going on in the world. How do we bring experts to explain the significance, whether that's a preprint publication or a major policy development? And I think that's been absolutely brilliant to help scientists have a voice and being a resource for, journalists. I think that's very positive. I think the one thing we really do need to look at is social media and how we do that. And I think, you know, on the one hand, you could say, okay, social media is just people chatting, you know, Twitter, whatever. But actually, going back to my earlier point about different groups and underserved populations, there are some groups who are very reliant on social media, and actually as industry, as researchers, as scientists, we need to find a way of engaging, because otherwise we're excluding populations that we want to engage with, and it might not be very comfortable, but we've got to find a way of doing that in the right way that's, you know, not there's just the right way of sensible information, but and it's a tough one, that one. And I think, you know, to your point about perhaps an inherent conservatism with a small c of organisations, again, it's a lot of organisations saying, no, we're not going to put something out on Twitter, and then you're going, yeah, but there's a load of disinformation out there, or just, and not malicious, just confusion, because nobody's engaging.
And I do think, to that point, sorry to interrupt you, but I do think also that in biotech, we tend not to have enough thought about the medical affairs side of stuff early on, because it'll suddenly hit you. And it's a good idea to be prepared. And interestingly, to what you just said, the Royal College of Child Health and Paediatrics, they have a TikTok site. So, because they believe that is how they get to their teenagers, and it's no good on anything else. They don't read Facebook, apparently. Well, I know that from my grandchildren. They think I'm very old-fashioned if I open Facebook.
I mean, social media works in different ways, doesn't it? I mean, what was really useful about it was the kind of public engagement on social media, and it was a really good platform where you could see people with different views debating about whether they should be vaccinated, and they debated in quite strong terms often, but it helped to encourage that engagement with the public.
I do think also it's a vehicle for us to educate people beyond when they're in medical school and so on, because actually we go on adult learning, and many things that my children and grandchildren learn in school are things that I had to learn as an adult. And I think we need to take adult learning probably more seriously, whether we do it through social media or something else. I think that was one of the big challenges for public health, was getting the public up to speed. I'm going to change tack slightly here and say, each of you, what do you think we could take from COVID into other either infections or some of the things that we talked about that are real big global health issues? Because I think that is probably one of the biggest things that we ought to be doing from COVID. So starting with you, Aiden, what do you think?
I'm going to repeat what I said earlier, which is really elevating the role of data and statistics to become a core part of decision making. You know, there's a common practice within clinical trials now, and I'll kind of describe it to you, and hopefully you will recognize part of it. You know, you might have a project team who are developing a protocol. They write all of the different sections of the protocol, and then they get to the bit that talks about sample size and statistics. And then they say, right, now we need a statistician to come and complete this section for us. So they engage a statistician, and then the conversation might go along the lines of, oh, and by the way, we have a budget which will allow us to run a study on whatever the number of patients is. And so your job is to go and come up with a rationale for this sample size. That's potentially an extreme example, it is common, right? Because the budgets have been raised before conducting the study. But, you know, it's clearly a nonsense, because any statistician, and we've already talked about, you know, the communications side, any statistician can go to the literature and trawl through it and come up with a set of assumptions that can be used to support virtually any study design.
So give me the answer you want and I'll
Yeah.
give you the rationale.
You know, so this is still quite common practice. I hear it almost weekly. That doesn't set a study up for success. That sets a study up to fail. You haven't de-risked the study at all. You know, so I think what has been useful during the pandemic is, one, greater awareness of some of the other tools that will be used, that can be used, such as modelling and simulation to de-risk studies. It's also been useful in the non-COVID space, because there were lots of studies that were delayed whilst COVID was going on, so they stayed in the planning phase for much longer. So in other words, the project teams invested a bit more time and effort and money on the planning piece, and I think that has set those up to be de-risked and set them up for success. So that for me is a big
I think
thing.
that's very important, and obviously underpinned by some of the things that June Rain is putting in place about doing modelling before you go to for your phase 1 and 2 studies,
Yeah.
which I think is really important. Ratko, what do you think we should be focusing on
Well,
that we've learned?
I spotted the 100+ trials with tocilizumab. I mean, you know, that's folly. It's sheer folly. So the question is, how do you coordinate trials? And there is only one solution. That's the WHO. But, you know, with, the US, Trump wanted to pull out of, well, he did pull out of the WHO, and various countries thinking they know better than the WHO, then obviously you have a big problem, because you have to give up some of your autonomy and sort of self-adoration, and put yourself in the hands of people who are, you know, out of your control. But I think there's only, you can only do these big things through big collaborative networks. So the speed of delivery would have been much better. Then how do you do the trials? Well, I think you raised something which resonated well with me, and which we're working on in Southampton, is to train all medical students to be able to, when they finish their medical studies, to be able to do research. You don't have to be the kind of researcher that I am, lab-based translational research. No, you don't have to. But just be able to do, simple studies. Then the way we capture data, I think, is probably the biggest challenge, but an enormous opportunity for IT geeks. You let them loose. So I'll give you one example. We did a study, and I unfortunately I can't mention too much about the details, but we did a study in a country where it's difficult to do research. But we did it with an app. A couple of colleagues, IT people, created an app they can put on their phone, and that's how that was their ECRF. The outcomes that we recorded were fairly simple. Need for intensive care. Did the patient actually go into intensive care? Pull down menu. You could complete it in literally 2 minutes on a ward round, which meant that they recruited at the time where patients were piling up outside the hospital, and they were able to deliver the study in 4 months. So, and of course, there are many potential problems with that. How do you QC the data? How do you monitor the data and so on? I'm not saying that this is the solution, but But it is possible, the technology is there, and then you can pull the data and that is of good enough quality and give it to statisticians to work.
So I think you raised two very important points there. One is that we shouldn't just be nationally focused. We must actually keep looking globally. And I think, you know...
It's a global pandemic, right?
Absolutely.
So,
and I think that we do, but maybe we, in terms of the research side, we should perhaps be a bit more coordinated with other people around the world. And I think some of that happened, but some of it has been quite difficult, really. Sheuli?
Yeah, so I think for me it's two things. It's about when we're designing that research, really thinking about different populations, which, you know, I'm sure over the years it's been said in lots of conferences, but it's absolutely been thrown into sharp relief. So how do we really design research and engage different populations so we can make sure that that research is applicable? And I think there's something else also around whether it's decentralised trials or taking trials to patients, but actually thinking about how can you get answers to questions without having to have patients coming into hospital either at all or minimising how much they have to come into hospital. So how can you use the technology or just different ways of thinking, and so that you can reach populations and get those answers. So those would be the two things in addition to, great comments there.
I think one thing which I think we've talked about lots of really positive things, and one thing just to finish off, and I don't really want to finish off on a negative note, but actually what is the biggest barrier to learning from COVID, and how do we resolve that? And I just sort of brief short answers, because we're running out of time, but what do you see as the biggest challenge, and what would be your idea to overcome it?
So just to be absolutely clear, the question is, what is the biggest barrier to
taking the learnings from COVID forward? That we've talked quite a lot of things about speed, about this. Take one of those things and tell me what is a barrier to that happening, and how can we overcome it, so that we come up with 3 or 4 things we can do, both in pharma and maybe in the public domain, that we can support moving forward. Do you want to start, Aiden?
Wow. That's a tough one. I mean, for me, one, I hope we don't revert to type, but I'm hopeful and pretty confident that the cat is out of the bag. So I think some of the things that we have got exposure to over the last 18 months will stick, because we've been able to try them out when we hadn't tried them before. So that's still a positive. I think there's quite a lot of work to do around the data access piece, and technology is going to be part of an answer to that. But, when I think about the wealth of data we have in terms of electronic health records, and there's a big challenge in getting access to those. That challenge hasn't gone away, but I think there's so much value in the data that enabling access to it, I think, is going to make a big difference that can be used for all development programs.
I think that access to data is a big one, has a lot of barriers, but there's lots of things we can do to get there, I think.
And Big Pharma themselves have enormous
loads of
data on programs that they have no interest in anymore.
Which we use for adverse events, we just don't use for, very good point. Ratko?
Yes, whilst I would prefix any suggestion with what I said earlier, that you have to do this analytically. You have to look at the whole experience and go through it very dispassionately to analyze what went wrong, what could have been done better. But my single biggest challenge is personal and collective ego, which throughout my career I've fought with egos, sometimes my own, often my own, in other words, we have to stop patting ourselves on the back, how we did well. Oh, didn't we do well? Yes, we did. Everybody's lovey-dovey. We have to park that and really, in all honesty, kind of look and take the good things and develop them further. But we must avoid making the same mistakes over and over. So
I think that's really important. It goes back to our point about collaboration. There is no I in teams. There is a me. If you're not listening,
there is a collective.
No I.
There is a collective. Jung said it very nicely. There's a collective ego. There's a collective phenotype. Psychological phenotype. So you can get institutions. Gee, aren't we good? We're good. Yeah.
Yeah.
We've got to be aware of the.
Sheuli, last,
So I think
not least.
is one of the issues will be just a feeling that people want to get back to normal, in inverted commas, talking about normal things and not talking about COVID, not talking about research. And I think that's then really up to us to basically translate what has happened in COVID from a health research perspective and say, actually, what does this mean for the future? So that rather than people go, oh, it's someone on BBC News talking about COVID research again, it's actually, no, they're talking about something that is future facing but is built on the learnings, whether that's knowledge about how you do research. At the moment, the population's got a great amount of knowledge about research, which is brilliant, but we're not going to be able to sustain that by keeping going with talking about the next COVID trial and the next one. We need to be able, as this community, to translate that into something people go, oh yeah, I do want to hear about that on the 10 o'clock news. And
yes,
and
especially, who's heard of the international stroke trial and things like that, which actually is so important.
Yeah, and how do we translate or translate that into stuff that will be interesting for the future and build on what we have now and not let people go, oh, it's COVID boring,
Yeah.
So.
Yeah. Aileen, we are 1 minute over.
That's what I was about to say. The camera has a burning question. That's not. Only one. There is one. Okay, I'm taking one. You were first.
You got the mic.
Yeah.
Hold on.
I'm going to do a double act.
Very good.
This is probably a question more for Aiden. So you were talking a lot about statistics being an important part of this equation. So can you explain to me the modelling process? Because the modelers and the simulations that were coming and the doomsday scenarios didn't really play out. There was a lot of interception and a lot of things that were happening at speed. But I'm just wondering, is there something fundamentally wrong that we need to be doing in designing these models and electronic health records and understanding patients in more detail?
Good question.
It's a great question. Well, the short answer is yes. I actually felt the models performed pretty well given the level of uncertainty around the kind of this scenario as it played out. We didn't understand COVID in the early days, and where models are really useful is to just evaluate different scenarios, the performance in different scenarios, what if type of scenarios, and we will never know the truth. What might have happened had we not intervened, had we not done the social distancing, the mask wearing or whatever. So we will never know the truth. But what I would say now, because we have a lot more information around COVID, that those models are going to be much more kind of robust. you know, but certainly there are two different bits here. One is the model itself, and is it a true reflection of what's going on? But then the other is the inputs to those models and how realistic are those. To me, it looks like the models themselves were pretty good. The inputs, there was so much uncertainty around those inputs that we really didn't know how grounded they were in reality. But it was still important to kind of run through the different scenarios and use those scenarios to guide the interventions that we took. And just one last point, I don't think it's useful to present sort of fantastic, unrealistic scenarios where, you know, you've got whatever the number was, 500,000 deaths, you know, because I think that undermines credibility. But I think the models themselves were incredibly useful for informing the kind of interventions that we needed to do. And I think it's also fair to say, without those interventions, the deaths would have been certainly a lot higher.
That's certainly true.
Okay, so Martin, if you can keep your question for the break to them, because I've already...
Yes, anybody can come and ask us questions.
Exactly. Just wanted to thank a lot of panelists and Flic, and you can join me in just thanking them. Not sure about working, maybe. Anyway, time for lunch, so enjoy that. A quick announcement. So IDX and Clinical Biology currently are running a naming an old competition, and for each submission they are giving to charity, so you can go see them, give it a try. So you may win all, and they will give to charity. So thank you.
Thank you. That was really useful. I hope that was all right.